Have you ever stopped to think that every single living thing, from the smallest microbe to the biggest whale, is basically running on a kind of biological software? Well, that software, that operating system, is genetics. It's the source code that dictates everything: how an organism is built, how it works, and how it passes its instructions down the line. Today, we're going to take a look under the hood of that code. This quote just nails it, right? It's the perfect way to frame this. Just like Newton's laws gave us this predictable, logical framework for the physical world, the principles of genetics do the exact same thing for the biological world. This isn't just a bunch of random chaos. It's a system. It has rules, it has logic, and it has outcomes we can actually predict. So, here's our game plan. We're going to start with those foundational Newton's laws of biology. Then we'll look at the physical code itself and how it gets passed on. After that, we'll get into the fun stuff, the complexities of how genes interact, and, what happens when you get bugs in the code. And finally, we'll connect all the dots and see why this is so incredibly important for engineers. All right, Section 1, Biology's operating system. Let's get right into those fundamental rules. So that brings us to Section 2, Mendel's laws. As we said, these are basically the Newton's laws of biology. Our story really kicks off with this guy, Gregor Mendel. He was meticulously experimenting with pea plants, of all things, and he stumbled upon the first core principle, the law of dominance. So he crossed a pure tall plant with a pure short one. And guess what? All of the new plants were tall, every single one. The tall trait was dominant, and it just completely masked the recessive short trait. It hadn't vanished, it was just laying low. But this is where it gets really brilliant. When he took those new tall plants and crossed them, that short trait popped right back up. This little chart, a Punnett square, shows exactly what happened. You get this perfect, predictable 3-to-1 ratio of tall to short plants. That silent trait was there the whole time, just waiting for its chance. This was huge. It proved that heredity wasn't some mushy blending of traits, but the passing of distinct, separate units. So how in the world does that happen? Well, that's the law of segregation. It basically says that for any trait, the 2 copies of a gene you have, one from mom, one from dad, get split up when sperm and eggs are made. This means the genetic deck gets a thorough shuffle every single generation, making sure every kid gets a fresh, unique combination. And it gets even more interesting. Mendel, he didn't stop there. He decided to track 2 different traits at the same time, like a seed's shape and its color. And what he found was that they're inherited completely independently. Whether a seed was round had absolutely zero influence on whether it was yellow. They were not a package deal. And this right here? This is the classic signature of that law. When you cross 2 traits that sort independently, you consistently get this beautiful 9-to-3 to 3-to-1 ratio. This is the key insight, really. It shows the incredible predictive power of genetics. This isn't just random biology, it's math. Okay, Section 3, the code and its transmission. So we've got the rules. Now let's look at the physical code itself, how we go from these abstract rules to actual physical reality. First up, a really key distinction. The genotype is the underlying genetic code. It's the ones and zeros, the source code, if you will. The phenotype is what you actually see, the expressed, observable trait, like the plant being tall or the seed being yellow. One is the blueprint, the other is the finished building. And that brings us to a super critical question. How is this code, this blueprint, actually copied and passed on? The whole system needs a totally reliable way to transmit its instructions, both for an organism to grow and for it to reproduce. The answer lies in 2 different kinds of cell division. Mitosis is basically biology's copy and paste function. It's for growth and repair, and it makes 2 identical daughter cells. But meiosis, that's for reproduction. It shuffles the genetic deck, creates 4 new cells with only half the genetic material, and that ensures all sorts of diversity in the next generation. So for Section 4, we're looking at when genes interact. Now, Mendel's laws are the foundation, for sure, but biology, it loves complexity. So let's look beyond those simple rules. A perfect example of this is something called epistasis. This is when one gene can pretty much hit the mute button on another gene. It doesn't matter what that second gene is trying to do. If the epistatic gene is active, it gets the final say. It's like a genetic override switch. A fantastic example is bee coloration. The final color of a bee isn't decided by just one gene,
but by an interaction. So you might have one gene that codes for a specific color, but another gene can act like an override, totally masking that first one and leading to a completely different outcome.
All right, Section 5, Errors in the Code. No operating system is perfect, and sometimes these chromosomal bugs can creep into the genetic code. And these bugs can happen on a pretty large scale, changing the whole structure of a chromosome. A piece of code can get deleted, or it can be accidentally duplicated. Sometimes a chunk breaks off, flips itself backwards and reattaches, or it can even get moved to a totally different chromosome. That's translocation. But the error doesn't have to be massive. Sometimes the bug is really small. It's in just a single gene. We call these monogenic disorders, and it's where one single faulty gene is the root cause of an entire disease.
And these single gene disorders, they follow predictable inheritance patterns. Some, like sickle cell anemia, are recessive, which means you need to get a bad copy of the gene from both parents. But others, like Huntington's disease, are dominant. Just one bad copy is enough to cause the illness. Understanding these patterns is absolutely crucial for tracking genetic diseases. Okay, let's bring it all home. Section 6, Engineering Biological Systems. We've gone through the rules, the code, the bugs. Now, the big question, why does any of this matter to an engineer? This quote really gets to the heart of it. The ultimate goal here is the rational engineering of biological systems. Think about it. Once we truly understand the rules of the code, we can go beyond just watching life happen. We can start to actively design it. We can debug genetic errors, optimize how cells work, and maybe even build entirely new biological functions from the ground up. Which leaves us with a pretty incredible thought to chew on. For centuries, we've been learning how to read the code of life. But now, the real challenge is learning how to write it.